Good afternoon. Please, Prof. Kate Walker, can you please stand? Prof. Eleanor, you stand next, but please wait. Okay. Okay. So, Prof. Kate Walker is a professor of obstetrics and a consultant obstetrician. Her research is based in the Nottingham Clinical Trials Unit at the University of Nottingham, and her clinical practice is at Nottingham University Hospitals Trust, where she divides her time equally between research and clinical work. Clinically, Professor Walker's interests are high-risk pregnancies, induction of labor, and labor ward management. Her PhD work examined induction of labor and stillbirth. Her research program has concentrated on randomized control trials in obstetrics, sexual health, and neonatology. She is the chief investigator for the GBS3, GLD, GILD, and DEBI trials. She also serves as a co-investigator on the recently concluded IVON-PP trial and the ongoing ADEN-V trial, both coordinated by CCTRIS. Prof. Eleanor, please stand. No? Please wait, we will still clap, just hold on. We will clap. Professor Eleanor Mitchell is a professor of clinical trials and global health from the Nottingham Clinical Trials Unit at the University of Nottingham. She is a methodologist with over 2 decades of experience in clinical trials and substantial experience in the design and conduct of large multi-centered randomized control trials, particularly in maternal and newborn health. She is the current co-investigator of 2 large publicly funded randomized trials in the United Kingdom, one focusing on gestational diabetes and the other on resuscitation of preterm infants. She's also chief investigator, of a large clustered trial in Kenya, investigating the use of an early warning score on hypothermia reduction in preterm infants. She has over 60 peer-reviewed publications and has published in prestigious journals, including the New England Journal of Medicine and the Lancet. She is the former chair of the United Kingdom's Trial Managers Network and is a member of national and international research funding bodies. She is also the Global Engagement Director of the School of Medicine at the University of Nottingham and serves as a co-investigator on the recently concluded, or served as a co-investigator on the recently concluded IVON-PP trial, and is a co-investigator on the ongoing ADEN-V trial, both coordinated by CCTRIS. Please, Prof. Kate Walker, can you stand? And please, can we all stand with a rising ovation? Let's appreciate these 2 great giants. Thank you.
Into mic. Would you use this? I use this. Can we give her another one?
Yeah. Hello. Good afternoon. It's such a great pleasure to be with you. I thought that my colleague who's from Nigeria was the happiest person I've ever met, but I've now realised you're just all joyful. What a wonderful country, what amazing people, and our visit is long overdue, Mercedes, and we couldn't be more happy to be here. So, just to give you an overview of what we're going to talk about, we want to just give you a flavour of some of the trials we do. I think we were talking on the plane here, both of us are involved in about 10 trials that are currently open, but we're just going to give you a flavour of one each to talk about some of the research that we lead. We also want to touch on studies within a trial, which is something that we're both very passionate about and focuses on trying to improve the methodology of trials to make them more efficient. And again, we'll give an example of that, of a study within a trial that we're doing within one of our diabetes trials. We want to talk about innovative trial design, Both of us sit on funding committees. I'm on a UK funding committee, Eleanor's on a Swedish one. And it's very clear that the direction of travel in the UK and in Sweden is these innovative trial designs. And as we've heard, that trials have to be agile, the staff working on them have to be flexible and agile, and that makes trials more efficient. And last, but actually probably more important than anything, we want to talk about collaboration and how important that is, and community engagement, which is something that Eleanor particularly has worked hugely on. So I'm going to start by talking about definitely the most difficult trial I've ever done, which is the GBS3 trial. And so many of you in the audience, I know we've got a real mixture in the audience, but many of you will have heard about Group B strep. So this is a bacteria that lives in our guts and our vaginas and helps keep those parts of our body healthy, doesn't cause any symptoms, and is not really of any relevance until women are pregnant and carry Group B strep. We know that a quarter of all women, up to a quarter of all women carry Group B strep, and when they're pregnant and they give birth, 50% of their babies will become colonised with the bacteria. Like their mums, most babies will remain well, asymptomatic from that colonisation, but about 3% of babies will develop an infection. And that's in the first 7 days of life, and it usually presents as sepsis, pneumonia, or meningitis less commonly. And unfortunately, Group B strep across the world has a huge burden of disease. So in a country like the UK, it's about 500 babies every year develop a Group B strep infection. And of those, 40 babies sadly die, completely preventable. And 25 babies are left with life-changing disability, almost always as a result of meningitis. In sub-Saharan Africa, that burden of disease is way higher, and that's partly due to higher colonisation rates in pregnant women in sub-Saharan Africa. It's also due to a lack of access to antibiotics in labour, which is our preventative treatment. And so there are over 50,000 infant deaths in sub-Saharan Africa, which are potentially preventable, just like in the UK. So do we have an effective treatment? Well, I hinted that we do, and that's antibiotics. So we've got good evidence for our effective intervention in women that we know are colonised with Group B strep. So 3 different trials have been done. They weren't massive, hundreds of women, in total 500 women. And for women known to be colonised with Group B strep, they randomised those women to either receive antibiotics in labour or not. All of those studies, unfortunately, were at high risk of bias, but the trials have shown that antibiotics in labour, which we give every 4 hours, reduces the risk of early onset infection from Group B strep in newborns by over 80%, very close to 90%. So how do we find out whether women carry Group B strep? Well, as I'm going to talk about, in the UK we don't test for Group B strep, but in many parts of the world women are routinely tested in pregnancy. And there's 2 different types of test. What's for a long time being considered the gold standard is taking a vaginal rectal swab. You can use the same swab, make sure you put it in the vagina first. That's sent to the lab. The swab is inoculated in a very rich broth to help the bacteria grow, and then the broth is plated out onto agar and the bacteria is grown. That result takes 48 to 72 hours. And it means we need to do it a little bit earlier, between 35 and 37 weeks gestation of pregnancy, in order that we've got a result before a timed birth. So the reason we do it as early as 35 weeks is because we time the birth of multiple pregnancy in the UK at 37 weeks. The other option, which has become much more commonplace since COVID pandemic, is a molecular test using PCR. And that means that we can do a rapid test in labour and get a result within 1 hour. So those are the two different types of tests. When we started the GBS3 trial, we weren't sure which was the best test, but during the course of the trial, GBS2 trial has published by my co-chief investigator for the GBS3 trial, and that shows that the rapid test is just as good as the gold standard culture test. So what do we currently do in the UK? I mentioned that we don't test for group B strep. What we do is we assess women for risk factors for their baby developing an early onset infection. And those risk factors are if your previous baby had group B strep disease, if you have incidentally found to be carrying group B strep, either in your urine or on a swab, if you are in preterm labour, and if you have a suspected infection during labour, normally which would be discovered with a high temperature. And if women have those risk factors, then we offer antibiotics. But unfortunately, nearly 70% of women who have risk factors don't actually carry the bacteria and therefore get these antibiotics unnecessarily. And of the women who don't have risk factors, actually 17% do carry group B strep. And so, unfortunately, of the babies who develop an early onset infection in the UK, 65% of them, their mothers don't have risk factors. So this approach, I hope you can see, is imperfect. And you might wonder why we follow this in the UK, and I'm going to explain why. So you can see in the graph on the left-hand side that the incidence of group B strep in the UK is on the rise. And in countries like the US, where they've adopted testing all pregnant women for group B strep since 1996, the incidence of group B strep disease in newborns has fallen. So that's just showing a comparison. We're not the only country that doesn't test, but 35 countries do offer universal testing in pregnancy. And they do that on the basis of observational data. So I mentioned that there's been trials of antibiotics for women known to be colonised with group B strep, but there has never, until the GBS3 trial, been a trial of testing for group B strep. So all we have to go on is observational data, which is at high risk of bias because women are not randomised, usually involves retrospective studies, and the studies vary widely. So they vary in the colonisation rate of group B strep in the different studies varies. The definition of infection varies and is often just based on culture positive, not clinically suspected infection. And so because of this, so there's been 3 systematic reviews of all of these studies. The best one is Haas-Verhoeven, which involved a systematic review of 10 studies over 11 million babies, so massive systematic review. And that showed that testing for group B strep in observational studies more than halves neonatal infections. And that's the basis for those 35 countries offering testing. But unfortunately, in the UK, we're very stringent, partly because we have a national health service. So healthcare in the UK is free at the point of demand for all women accessing care during pregnancy. Your care is completely free. And therefore, we have to be sure that screening or testing that we do in pregnancy is both clinically and cost effective. And so this is the criteria that the UK use to decide whether they're going to adopt testing for anything. The condition's got to be important. Well, we can see it is. It causes death and significant morbidity. We've got to have a simple test for group B strep. We've got 2 of them. We have to have an effective treatment, which we do with antibiotics, preventing over 80% of infections. But this is what we don't have, evidence from a randomised trial. Therefore, we don't know whether the benefit of testing in reducing infection in newborns outweighs the harm of testing, which is that potentially many more women need to receive antibiotics in labour. That can cause harm for the mother, rarely, anaphylaxis in the mother from IV antibiotics, but it also can have implications for the baby, and we know that antibiotics given to the mother changes the baby's microbiome, their gut microbiome, and that could have potential implications. We, I'm glad to say, have been able to address the next point because we've done alongside qualitative studies in the GBS3 trial, which shows that testing for GBS is highly acceptable to both healthcare providers and to patients. And in the trial, we will determine cost effectiveness as well as clinical effectiveness, but at the moment, that is very uncertain. So, 5 years ago, we designed the GBS3 trial, and we have concluded the trial. We're in the analysis phase now, hoping to publish in March 2026. It's a cluster randomised trial. So rather than individually randomise women to testing or no testing, we randomised hospitals as the unit of randomisation, and every woman receiving pregnancy care in that hospital had the same thing. Our sample size was an eye-watering 320,000 women, and I'm delighted to say that we've got 317,000 women in the data set, which is being analysed. Our setting was 71 of our 150 maternity units in the UK, and as I say, we've completed recruitment for the study. So our population was all women having a vaginal birth or a trial of vaginal birth. Obviously, some women will end up with an emergency caesarean beyond 24 weeks gestation. And our intervention, because when we designed the trial, we didn't know that the point of care test was as good as the longer-winded culture test, we set out to determine what type of testing was better as well. So our intervention is both the culture test or the rapid test. Our control group is our usual care, this risk factor-based approach, just assessing the mum for risk factors, which, as I've shown you, is imperfect. And our primary outcome was all-cause early neonatal sepsis. So why did we choose all-cause? Well, we know antibiotics, although group B strep is the most common cause of infection in newborns, we know that antibiotics in labour reduces the risk of early onset infection from other pathogens. So we thought it was very important to capture that. And we wanted to include both culture positive sepsis, which is bacteria in a normally sterile site like blood or CSF, with clinically suspected sepsis, and this is an internationally agreed definition of clinically suspected sepsis. So how did we do a trial of 320,000 women? Sounds very pricey for the budget. So one of the most important things to make this trial affordable was that we utilised routine data. So rather than collect individual data from every woman, one of those 320,000 women, we use data that's routinely reported by hospitals to various different bodies. So unfortunately, all of the data doesn't go to the same place. So every woman who gives birth in the UK, we send maternity data to the Maternity Services data set. Every woman who has a positive culture, we send that culture result to the UK Health Security Agency. Every baby that's admitted to neonatal unit, we send the data to the neonatal research database. And every baby who's sent home but gets readmitted to paediatric intensive care, we send that to a network for PICU. So we collected all this data for every single woman who took part in the trial, and then we're merging that data together. That's what's currently happening in order to get all of our outcomes for the trial. The other thing that made it affordable is that because it's a cluster trial, we were able to only take verbal consent. So women in the trial don't have to sign a consent form, which is typical in most trials. All they have to do is verbally agree to a swab. Now, that was possible through our community engagement, which I will talk about in a minute, which allowed us to get this CAG approval, confidential advisory group approval, which is pretty difficult. You have to prove that it's just not possible to get individual consent, that it's ethical to not get informed consent, and you need a lot of community engagement to convince the CAG committee that you're doing the right thing. The cluster design also helped because it meant we could rely on clinical care teams to deliver the intervention. So we trained our clinical care teams in the 71 hospitals, and they either did testing or they didn't. We didn't need a member of the research team to be able to administer the test. Ooh. And so 71 maternity units were randomised into one of three groups. Half of units were randomised to usual care. My own unit in Nottingham, we were very disappointed, but that's what we got randomised to. 18 of our sites did the antenatal culture, and 17 adopted the rapid test. We had a very collaborative approach. We do have a collaborative approach for all of our trials, but particularly for this one because it was so big. So we needed a health economist, but we also needed a health economist who specialised in perinatal trials, and that person was from Oxford. We needed an academic midwife who'd got trials experience, and that person was in the University of Lancashire. We needed qualitative researchers who'd done perinatal studies, and they're in the City of London. We had a neonatologist who was based in Canada, and we had a microbiologist who'd always done Group B strep research based in Birmingham. All of those different healthcare providers came together to form the team. And crucially, we had community engagement from a charity that works to prevent Group B strep disease, Group B strep support, but also a charity that just helps pregnant women and helps with antenatal education. And that's quite commonly the way we do our patient and public involvement, that we will get groups that look at the specific condition, but also groups that just represent pregnant women. We had patient and public involvement co-applicants right from the very start to design the research. We conducted focus groups with women and then had an ongoing Facebook group, both for women who'd had a previous baby with Group B strep and women with uncomplicated histories. And we did regular consultation throughout the trial and ongoing now to help with making plans for dissemination of the research. I'm going to hand over to Eleanor.
Thanks, Katie. And just to extend our gratitude for being invited today. It really is wonderful to be talking to you, having worked with Besady and her team for some time now to be here in Nigeria. So thank you very much. So Kate's talked about the GBS3 trial, which utilises routine data, and that's really one of the great innovations within the UK about how we've been moving clinical trials forwards. It's only possible because we've got an excellent infrastructure in terms of routinely collected clinical data, but increasingly those sorts of trials are being done because they're considered more efficient. They can be done more quickly, they're more cost effective. The trial I'm going to tell you about now is called the GUILD trial, and it's a trial that Kate and I are the co-chief investigators on in pregnant women with gestational diabetes. So in the UK, gestational diabetes affects around 8% of pregnant women. And we know that controlling maternal blood sugars throughout their pregnancy is really important, but it's particularly important during their labour. And that's because of the impact of high blood sugars through to the baby. So when the woman is during labour, her insulin levels can increase, insulin is given, and the baby's insulin levels are therefore quite high. And when the baby is born and the placenta, the cord is clamped, and that placental blood flow stops, the baby can have a really big drop in their blood sugars, leading to neonatal hypoglycemia and potentially admission to the neonatal intensive care unit. And so controlling a woman's blood sugars is really important, both in terms of outcomes for the mum and outcomes for the baby. And at the moment in the UK, women with gestational diabetes have what we call tight glucose control during their labour. So their blood sugars are monitored very frequently, they're kept in a very tight range, and that might be appropriate for women with type 1 or type 2 diabetes, but we think it might be safe to do something a little bit more relaxed for these women. We need to make sure that when we're monitoring blood sugars in labour, that we do this in a really evidence-based way. And at the moment, there's just a lack of evidence to say that you actually need to monitor so tightly in this particular group of women. We know that intensive monitoring can then lead to an increased use of insulin, and that can then obviously make the mum's blood sugars go the other way. What we want to do is look at a more relaxed blood sugar control monitoring. And we think that that could reduce overtreatment for the woman during her labour. It might improve their birth experience because they're not constantly having their fingers pricked and being tested. And importantly, in any health system, it could reduce midwifery workload. But what we don't know is if it's safe for the baby. There could be a risk that it could increase the risk of neonatal hypoglycemia and admission to the newborn unit. And that's what stopped people having this more relaxed approach to glucose control in the past. So that's where GILD comes in. So we're running a multi-centre, open-label, randomised controlled trial, and this is a non-inferiority trial. So we're not suggesting that more relaxed control is better than tight control. We don't think that's possible. But what we are suggesting is it could be just as good as tight control. And that's what a non-inferiority trial means. A non-inferiority trial is based on there being a trade-off. So we're considering it could be just as good from a safety perspective. It may result in no more neonatal hypoglycemia or admissions to the NICU, but actually women could have a much better birth experience. It could reduce midwifery workload. It could be more cost effective. So we're running this trial in 23 hospitals around the UK, aiming to recruit over 1,600 women, and we're comparing more relaxed and tight control. There's our definition. So our intervention, which is the more relaxed approach, is we're testing blood sugars every 2 to 4 hours and keeping the levels between 4 to 10 millimoles per litre. And we're comparing with the current standard care in the UK, which is hourly testing and keeping between 4 to 7 millimoles per litre. I'm not going to go into the eligibility criteria here, but you can see them on the screen. But we're recruiting women who are pregnant with a single baby and who are planning to give birth at term. And we've got a co-primary outcome. So there are two things that we're really interested in here. Aiming to look at neonatal hypoglycemia and admission to the neonatal unit. And actually, as we're applying for funding for this from the National Institute for Health Research, Kate and I wanted to do this trial to look at neonatal hypoglycemia, and the funders kept asking us to also look at neonatal unit admission, and we said no 2 or 3 times and then realised they wouldn't give us the money unless we agreed. So we did say yes, and we've got a co-primary outcome. We've also got several secondary outcomes focusing on neonatal and maternal clinical outcomes, but we're really interested in acceptability. We're particularly interested in the experience from the mum during the birth and what she thinks to the approach of being monitored more or less frequently, adherence to that strategy, and, as I've said, cost effectiveness. So a few examples of how we're raising awareness about the trial. Just like you, when we've been around the hospitals and we've seen your brilliant publicity of the ASIN-V trial, we've also got lots of posters in our hospitals. Many of our women are managed in the community by community midwives, and so it's really important as our wider engagement to work with community midwives, and we're providing information to them about the trial. And we spend a lot of time making our information look attractive, rather than just providing kind of standard information sheets to try and engage with patients. And our community engagement work that we've been leading has really helped get some of this right. So we've not been doing this trial for long. It's reasonably early in its recruitment. We've open to recruitment in 8 out of 23 hospitals, and you can see there the number of women who have given their consent and randomised, and 8 of them have given birth. The map on the screen shows where our sites are based around the UK, and the graph shows our recruitment so far. And recruitment in trials is really difficult. If you've ever been involved in a clinical trial, recruiting patients is probably one of the hardest things to do. And so Kate and I are very pleased that at the moment, our target recruitment is in line exactly with our predicted recruitment. So far we're getting it right, and we hopefully want those red and black lines to continue absolutely as they are to the end of the trial. Oops. So that's 2 examples of the big perinatal trials that we're doing at the moment. But at the University of Nottingham, we're also really interested in about how we do trials. As I've said, they're very difficult to do. And so one way where we can try and think about how we do trials is to embed methodology studies into trials. And again, this is an increasing and emerging area of research, particularly in the UK, lots of groups leading this. So trials are complex and time-consuming, and it's quite remarkable that really clinical trials are there to find out the best evidence for a particular intervention, to provide best evidence for healthcare. But actually, there's very little evidence on how we do trials. We don't really know, and we may make lots of things up as we go along. We basically try things. If they work, we'll keep going with it. If they don't work, we'll try something else. But it's not very robust. So trials methodology is all about finding out what really works, finding evidence to see how we design and run our trials. And that's to make them more efficient, so that ultimately we can get the results of our trials to the people that matter the most, the patients and the health professionals who serve those patients. Any aspect of trial design or conduct can be considered when you're thinking about trials methodology. And some projects are conducted entirely standalone. So, for example, I've got a PhD student at the moment who's looking at how do we market our trials to patients in the best possible way to improve recruitment. But you could also do embedded methodology studies in existing trials, which I'll tell you about in a moment. So a study within a trial, or a SWOT, is an increasing study type that are being done in the UK. And actually one of our main funders, so we get a lot of funding through the research part of the Department of Health and Social Care in the UK. They are increasingly happy to fund these methodological studies because it's in their interest. They invest millions of pounds in clinical trials, and it's in their interest to make the trials more efficient, done more quickly, and cost less money. So a study within a trial has emerged probably over the last decade or so, and there are some leading groups in the UK. So the Trial Forge Centre, who are based in the University of Aberdeen in Scotland, and a group of researchers based in Northern Ireland in the UK. And they've provided some great guidance on how these types of studies should be done, and importantly, a repository of all the ongoing studies. So just like when you register a clinical trial to be transparent and tell the world that you're doing a clinical trial in a particular area, there's also a repository of these methodological studies. And that's important to avoid research waste. We shouldn't be doing the same types of research studies over and over again. We should be focusing on innovation. So we've embedded a SWOT within the Guild trial, and this focuses on making sure we recruit the right women to our trial. So South Asian women are disproportionately affected by gestational diabetes compared to white European women. Yet in the UK, there are many South Asian women, but they have traditionally not been involved in research. They've been underserved by researchers. We've not done a great job at being as inclusive as we can be in our trials. And so we've designed a package that we hope will increase participation of this group of women. And we've been working with community groups to help get that right, doing lots of focus groups. We've designed some cultural awareness training for sites. We've designed special recruitment materials that focus on different aspects of gestational diabetes that South Asian women have told us are important. And we've appointed 3 women who have had gestational diabetes who we're calling community connectors. And they do exactly that. So if a woman who is considering participating in the trial has spoken to the doctor and the research nurse, but may have some questions and feel like they might want to talk to somebody to get some impartial advice, they can connect with a community connector who can provide that ad hoc peer support. So we're doing this SWOT to then see, can we recruit this group of women into the trial? So half of our trial sites will have the inclusivity package and half won't. And we'll make some simple comparisons at the end to look at the recruitment rates. So I'm going to pass over back now to Kate, who's going to talk a little bit more about some of these innovative designs.
Thanks, Eleanor. So, we've talked about some of these designs already, so pretty much everyone in the room, I suspect, will be familiar with a parallel design. That's, we're doing that in Guild, so we get a group of women from one population. In Guild, it's women with GDM, and women are individually randomised either to tight control or to less tight control. We follow both groups up, those groups run in parallel, hence the name, and at the end we conclude, is the primary outcome more common in one group than the other? And in most parallel trials, that's talking about individual randomisation. We've also heard that in the GBS3 trial, we're doing cluster randomisation. So rather than individually randomise women, we randomise by the hospital that the woman's giving birth at. Another design that we did just want to mention, because it's very popular and we're always getting asked by people, should we do a stepped wedge? So we thought it was important just to include it, even though it's not innovative. And a stepped wedge trial is where the intervention is started at different time points in the different clusters or in the individual patients. And stepped wedges in general, there are lots of different sort of designs of a stepped wedge, but this is the most common, where all clusters start in the control phase and at different intervals get randomised to the intervention, and that's randomly allocated. Stepped wedges are popular because they allow especially when it's difficult to implement the intervention at every single site at one time point. So if you imagine, we're doing a stepped wedge trial called Starship at the moment with University of Manchester, and that's looking at the Fetal Medicine Foundation algorithm for preventing preeclampsia. Part of that includes doing a special first trimester blood flow measurement to look at the blood flow to the womb, which is not commonly done, and therefore we have to train every sonographer at every hospital before we can implement the Starship trial, and therefore a stepped wedge is the perfect design for that trial. It's also appealing because every single site gets the intervention at some point. And actually what's really difficult is persuading sites to join if they're never going to benefit from the intervention. So I mentioned earlier that in the GBS3 trial in Nottingham, we were randomised to usual care. We never got to do a GBS test, which was hugely disappointing. So beauty of stepped wedges is that every hospital who takes part for a certain finite period of time will get to do the intervention. But the downside is that they're really complex, and logistically they're a nightmare, because you have to be ready to randomise sites pretty early on, when normally you're sort of getting sites set up gradually during the first 6 to 9 months of a trial. There's also an issue if it is individuals being randomised, that there can be contamination. So that's a really important concept in clinical trials, where, so because a woman who, I'll give you an example, there's a trial that's being done in women with anorexia, very severe eating disorder, and they're comparing high calorie diet with a lower calorie diet to see if there's an increase in refeeding syndrome. The concern is that if people with anorexia in the same ward are receiving different nutritional supplements, whether there'll be conversations and they're realising differences are happening, or staff see that one woman is doing better from her high calorie diet, and that might influence the practice of doctors and nurses on the eating disorder ward.
Thank you. I'm going to talk a little bit about this concept of using a master protocol, but before I do, we're talking about innovative trial designs here. I want to kind of encourage anyone that's considering doing a clinical trial now or in the future to use the most appropriate and the most simple design that you can to answer the question robustly. So the parallel design has been around for a long time. They're still hard work to do, but actually if you can answer the research question that you're interested in using a parallel design, you don't need a fancy trial design. You don't need to do anything innovative. It's been around for a long time because it's a decent design. Okay, so you might hear some of these designs that I'm going to tell you about now and think, oh, I'd really like to do that type of trial. And that's great if that's the best design for the question that you're answering. But I have had people approach me saying, we really want to do this type of trial, and actually there's a much simpler approach to doing it. And trials are hard enough as they are, so we shouldn't make them any harder. So a master protocol essentially answers multiple questions using a single protocol, and that could make it more efficient. So we've been talking about saving time, and that might be because you're sharing documents. You've not got so much paperwork in clinical trials. You want to try and minimize that. So sharing documents is very helpful, and sharing resources is also really helpful. This diagram here just shows you some different types of design that all utilize a master protocol design. dependent on whether you're interested in multiple diseases or populations, multiple interventions for a single disease or population, and whether you're planning on designing the trial from the outset and sticking to it or adapting that design as you go. So we're going to talk a little bit about trials called basket trials, umbrella trials, and platform trials, which also confusingly can be referred to as adaptive or MAMS, multi-arm, multi-stage. So this is a figure taken from some work led by Park about 5 or 6 years ago, and they did a systematic review which looked at the number of trials that were utilising this quite innovative master protocol design. And you can see there that over about 20 years there's been a massive increase in the number of this type of trial. So the white bars show the basket trials, the grey, the umbrella trials, and the black ones is the platform trials. Basket by far are the most popular designs that have been utilised using this type of protocol. So I'm going to talk a little bit about the basket trial. So that's where you have multiple diseases or populations, but you're using the same intervention, and you fix your design at the beginning like you would most trials. This definition here I really like, and this is the one that's been described in the Park paper that I just mentioned to you. So a basket trial they defined as any prospective clinical trial that tests the utility of an intervention. So they mean either the effectiveness, the dose of a particular medicine, or safety in a study population of multiple diseases. There's got to be something common between them, so common predictive biomarkers or other common indicators or patient characteristics that could be used to predict whether a patient will respond to a specific intervention. And although basket trials are we think of them as quite new, actually the first basket trial was done back in 2001. But it's fair to say you could see from the previous slide that they have increased massively over time. Most of the trials in the Park review were drug trials that were basket trials, and a lot of them weren't randomised. So increasingly this is being used, and I think as kind of methodology becomes more advanced, people are learning more about how to use these designs. If you're considering a basket trial design, you've got to have a strong rationale mechanistically of why that intervention could be used in multiple diseases or different populations. You need to think up front, how are you going to organise your participants? So who are in your baskets? And in some populations that might be very easy to define, and some it may be a bit trickier, and it could take some time to get that right. You should be standardising your eligibility criteria, but also being flexible enough to make modifications for the different groups of participants. And from a statistical perspective, you'd usually analyse the data separately, so for the different baskets, and you'd power them separately. There are some statistical techniques that can be used where you can kind of borrow some power from each group, but mostly baskets are considered separate. And you should make the processes as simple as possible for sites in any trial. But also when you're doing a more complex design such as this one, it's particularly important to explain the trial really simply to the recruiting sites and providing clear training. So the Otter trial is a trial that I'm a co-investigator on in the UK, and this is a trial that we're conducting in 2 populations. We're interested in looking at the effect of omeprazole, so a common drug that's used in these babies, and comparing that with a matched placebo to see if omeprazole reduces symptoms of gastroesophageal reflux. Omeprazole is used all the time, but there's very little evidence in babies that it's actually effective, and we don't know if actually it could cause more harm. So we've designed a basket trial because we want to find out the answer to this question in term and preterm infants. And you can see there the description of the population. It's broadly similar, as I was explaining in the previous slide, but there are some modifications dependent on whether they're a term or preterm birth. So we are in the process of setting this trial up at the moment. We'll be recruiting from 10 primary care sites and 35 hospitals around the UK. And it's quite complex. There's recruitment in lots of different places in this trial, which makes it challenging. So we're recruiting from neonatal units and paediatric wards, but also from emergency departments. You know, parents get very stressed when they've got a crying baby who's been crying for a long time and often present at the emergency department in desperation. So we will be recruiting from ED and also outpatient clinics. We need 500 babies in total, 200 in each population, and then we've accounted for some parents kind of becoming lost to follow-up throughout. So we need 500 in total, and there's just a link there for more information for anyone who's interested. So umbrella trials are a little bit different. So rather than testing an intervention, a single intervention in multiple populations, it's the reverse. So we're actually looking at a single population or single disease, but testing multiple interventions. And again, you'd fix that design up front. And this is the definition used in the Park paper. I'll just leave it there for a moment so you can read that. I'm not going to go into any examples here, and that's deliberate. Firstly, there aren't lots of umbrella trials being conducted. They're predominantly in patients with cancer, where there may be multiple treatments used for a specific type of cancer. We don't have experience at the University of Nottingham in conducting umbrella trials because we don't do cancer trials predominantly. But if that's something you're interested in, where you're looking at lots of interventions that could target a single type of cancer, for example, you might want to consider utilising an umbrella design.
So the only trial design that people ask me more about than stepped wedge is platform trials. Everybody wants to do a platform trial. So why are they so popular? Well, going back to your comments earlier, Deputy Provost, about agility, these are the most agile of trials. So you're studying in a platform trial, in general, a specific disease, which is a bit problematic in pregnancy, and you evaluate multiple different interventions, and the trial is agile, so you can drop interventions if in an interim analysis that intervention doesn't look like it works. And you can add interventions, either if new interventions suddenly become available on the market, or if you've dropped an intervention so you have the resource to be able to test another one. They're master protocol trials, just like Eleanor's talked about, and they're efficient because they have a common control group. So the same control group is used for comparison with every different intervention arm in the trial. And in general, they're open-ended, so you have to be able to have a pipeline, which is the most common word associated with platform trials. You have to have a pipeline of ideas that you're going to be able to test over a period of time. And again, that makes it a bit problematic for us in pregnancy, because our control group, the recovery trial, was responsible for showing that dexamethasone reduces mortality from COVID. It went on to show 3 other drugs reduce mortality. And actually, just as importantly, it showed that these 8 interventions don't work and don't reduce mortality from COVID. And the recovery trial is now still being used for pneumonia. I have to say, it's important to say that partly why the recovery trial was such a big success is all the rest of our research in the UK was put on pause. So we paused GBS3 trial for 1 year, and our entire research infrastructure recruited to recovery. So my research midwives, who would normally be recruiting to perinatal trials, were completing outcome data collection for COVID patients who weren't pregnant. We put all of our eggs in one basket and delivered the recovery trial. It's hard to know outside of a pandemic whether the trial would have been as successful as it has been, given that. So what are the advantages of platform trials? Well, you get quicker results. You find out whether treatments work more quickly, and that means they're cheaper, because even a month longer duration has a significant impact on the cost of a trial. So anything that makes it quicker makes it cheaper. The disadvantage, as we have learned when we worked up a perinatal platform trial, is that they're very complex. And actually the governance can be very complex because they're ambitious and broad, and so you need lots of you need resource from more than one clinical trials unit to be able to deliver a platform trial. And particularly for our field of pregnancy, unfortunately, they're best suited for a single disease state, which makes it quite difficult to do in the perinatal field.
Okay, so throughout the welcome remarks, there was lots of mention of collaboration, and that's so important in clinical trials. So we're going to mention why that's important now, and this concept of team science. So you might think it's a bit weird that I've got a picture here of a small child cutting his father's hair. Now, I don't know about what happened in Nigeria during the pandemic, but when everything shut down in the UK, people started cutting their own hair, okay? And as you can see from this photo, they didn't do it very well. Now, clinical trials need to be conducted by people who have got the expertise in conducting clinical trials. You know, we go to the hairdressers or the barbers to have our hair cut because they've been trained and they have the expertise in doing it. And look what happens when you try to do it yourself. As we've said throughout, trials are complex, take a long time and cost a lot of money. And so many trials are done that are not good quality. And that's unethical. It's unethical to the patients. It's a waste of money. It's a waste of resource. And overall, it contributes to this concept of research waste. So if you're going to do a clinical trial, you really need to work with the most appropriate people who have got the expertise to conduct the trial. So I don't know if any of you have come across this concept of team science. This is a definition by NIH in the States. But team science is a collaborative effort to address a scientific challenge that leverages the strengths and expertise of professionals, often in different fields. And this definition goes on to say that although single investigator approaches might be appropriate for some endeavours, actually, if you've got coordinated teams of investigators with different skill and knowledge, that's what you need to tackle these big societal problems. Clinical trials absolutely epitomise team science. So long gone, I hope, are the days of a single doctor trying to do a clinical trial. You know, that's what has happened in the past. And I'm really pleased to say that that's probably a thing of the past. And actually trials are being done by these big teams. So who should be in your team? Of course, depends on what the trial is about and what the question is that you're answering. I'm not going to read all of these, but you can see here, it's a big multidisciplinary team that you need, including people who are trained clinically, methodologists, and also people who have got expertise by experience. So working with patients and members of the public. I don't mean in terms of them participating, I mean in terms of them engaging in the design and the conduct of the trial.
And that leads us to talk about community engagement. So everything I've learnt about community engagement, I've learnt from Eleanor, and I'm not sure why I'm delivering these next few slides, because she is the expert in it. In the UK, we call it patient and public involvement, but I know it would be more commonly called community engagement in this setting. And what we mean by patient and public involvement is that the research is carried out with or by members of the public rather than carried out to them or for them. And it's about an active partnership between researchers and the patients that we serve. Engagement is about making sure that information and knowledge about the research is disseminated to the public. It's not about participation in trials. It's not about recruitment and making sure that you're getting those graphs looking beautiful by hitting your recruitment targets. And patient and public involvement or community engagement is absolutely central to every single trial that we do, and it's completely fundamental to making sure that the research we do is the right question, that the design is right, and therefore that the trial is going to be successful and make an impact. And we've just chosen two examples. We do patient and public involvement in every single trial we do, but I particularly wanted to choose this example, which is not a trial, but it's a really interesting study about trying to promote physiological birth. So physiological birth is awaiting spontaneous onset of labour. It's about avoiding interventions during birth. It might be allowing a physiological third stage where you wait for the placenta to separate naturally without using medication. And I think we could learn a lot from you in your setting about physiological birth, but in the UK, physiological births become almost unheard of. And actually there's lots of women wanting a physiological birth, and we need to make sure that women get choice while also keeping them safe, because there's some downsides to a physiological birth. And this is the CCTRIS study, and to me it's been absolutely extraordinary because it's moving beyond engagement with the public to the public doing the research. So you can see from that pie chart, the lilac colour, over a quarter of the budget for the whole project was funding patient and public co-applicants to not just input into the study, but to do the study. And so they gave us this input right from the start, a team of them, so that there was a balance in the team. Rather than having one PPI lead, we had a whole group of them. They've been involved in research activities, so when I'm selecting papers for a critical interpretive synthesis, so are the members of the public, because we know what we choose to include is going to impact on the results of the study. And they didn't just take part in stakeholder advisory network meetings, they organised them, they ran them, they delivered them, and so they were the best PPI network events I've ever been to, because they were led by members of the public, and they either co-wrote or one of the publications they've written themselves. So a real completely different perspective on PPIE, and it's been a real eye-opener, but actually there have been some hurdles. So one of the big hurdles was that initial grant development. There's no funding. And you're expecting people, you know, I'm paid a salary, so when I'm writing a grant, my time is funded, but you're expecting people to give a big portion of their time for free. And that's what needs to change if we're really going to make this a reality for other projects.
Nope. So the other example that we're going to share is from the RAPID programme. So most of the time when we design trials, the research question is agreed by usually the doctor. So they've got an idea of something that they want to test, and we work together and we design the trial. But it's a research question that's of importance to the health community. The RAPID programme has turned that entirely the other way, and what we're doing is asking patients what are the most important research questions. And they're quite different to what some of the health professionals think are important. We're then working with members of the public to then co-design trials that answer those questions, co-design the interventions that will be tested in those trials. And the 3 trials that are involved in this programme are very simple interventions and questions, but are really matter to patients who have got lived experience of eczema. They want to know, how often should they have a wash when they've got eczema? How often should they use their topical steroid cream for? What should they do to try and stop the itching when they've got eczema? These trials are embedded within the programme, and this isn't using an innovative approach, doing an entirely online trial. There are no recruiting sites, no hospitals involved. It's entirely decentralised. We advertise the trial on social media and through charities and support groups. People sign up online, they answer the questionnaires online, they provide their outcome data online, and then we quickly analyse the data at the end and disseminate the results to participants. So that's an example of a really innovative programme where we're working with members of the public.
So, just in conclusion, we've talked about 2 different trial designs that we've been involved in. We've talked about innovative trial designs like platform trials and basket trials. We hope we've encouraged you to think about studies within a trial to test methodology and make trials as efficient as possible. And really crucially, we've talked about collaboration. You've seen that in the examples that we've given, and about patient and public involvement. We just want to thank you once again for the invite. Thank you for the warm welcome. Thank you for hosting us and giving us such an amazing experience. We want to thank Nottingham Clinical Trials Unit, because all of the things we've presented are only possible through a massive team effort from a huge multidisciplinary team. And we want to thank our funder, the National Institute for Health Research, who have funded both of the trials that we've talked about.
And we're going to finish with a shameless plug. So we're both really interested in growing new researchers and increasing research capacity in the UK and in other countries. And so I'm the director of a suite of courses within the Nottingham Clinical Trials Unit, where we teach people how to design and run clinical trials. So we run introductory courses into trial design and conduct, and some of Mercedes' team have joined those courses. We're now doing courses that are exploring qualitative methods in clinical trials. We offer in-person and online delivery, and we offer discounted places for researchers who are based in low and middle income countries. And this is something that we're really passionate about. I think somebody in the introductions acknowledged the importance of having a pipeline of sustainable researchers for the future. So the future are the people who are learning, studying medicine now, studying kind of different approaches into trials. And doing high quality trials is absolutely key, and for that you need training. So do consider the programme if you're interested. So thank you once again, it's been a delight. Different approaches into trials, and doing high quality trials is absolutely key, and for that you need training. So I think we have a little time for questions. So thank you once again. So I don't know how we're going to do this.
Thank you, everyone. So we're going to, if you have a question, please raise your hand so that we can take all the questions. And also for those online, please send in your questions so that we can take note of them. So admin, can you please, if you could just signify to raise your hand so that, yeah.
Thank you, Professor Helena and Professor Kate. I think that is quite wonderful. I think they deserve a round of applause. I think just like you are going through the presentation, I was getting a lot of messages from online audience telling me that they are learning quite a lot, and I'm sure this will not be the last time we are going to see you around, and we'll still have the pleasure of getting to learn quite a lot from you. My question is actually maybe not a question, maybe just a clarification. Is with regards to the emphasis we laid on community engagement, which of course we call the public involvement engagement in the UK in clinical trial. I notice that the emphasis is being laid by a lot of funders now on the need to involve the public in designing your trial. I just want to know from your experience, because it looked a little bit theoretical to me sometimes, especially at the science stage, which you have quite alluded to, that where is the fund to actually involve the public when you want to design your trial? So how do you navigate that? Where do you get the fund? Do you have to tell the funder that you need money to be able to do this? Or probably maybe your institution help you in that regard? Maybe it's something we'll start asking Professor Afolabi to do also in CCTRIS. Thank you very much.
Thank you very much for the question. So our university does have small pockets of funding. They're very small. They're normally a couple of £100, but they're enough to hold 1 or 2 meetings, and they're enough to be an incentive for people to join. So that there are other sources of funding, but we tend to go locally just because it's quicker. We also have lots of different ways that we reach out. So we have Eleanor has a very successful perinatal community hosted on Facebook called Bump to Baby. So we're able to reach out to that group and say, has anybody had gestational diabetes? Would you like to come and help us design a project? We utilise social media to identify people, and we also use our charity partners. So going back to that, patients with the disease, patients without the disease, we tend to use those 2 groups, and they're often very successful. But don't forget, many of you are clinicians in the room. The beauty is that you just have ready access. So I'm constantly asking my own patients, could I just talk to you about this trial? What do you think? Would you join? Would you have any problem? So you have ready access to patients, which is what PPIE is all about. Anything to add?
Thank you very much. I don't think I've ever witnessed a well-coordinated twin presentation of this type before. It is fantastic. And I want to thank you, the 2 of you, for the knowledge I gained when you said master protocol design. I haven't heard of that before, and I'm happy I'm in this class. Now, master protocol design, the parallel, the cluster, the step wedge. Where is the one that we call switch over kind of a design?
Yeah.
That's just one question.
Can I take that one first?
Okay, I hold on.
Well, first of all, thank you very much for your kind comments. Kate and I are laughing now because of the question. We had some slides in there about a crossover design. So if you're wondering what a crossover design is, it's essentially where you randomize to an order of treatment. So you have 2 groups, and patients start with treatment A, they have a washout period, and then they have treatment B, and the other group will do it the other way around. The reason we didn't include it is because the design has been around for quite a long time, so it's not particularly innovative, and they're quite difficult to do actually, because they're only relevant for very specific disease areas because of needing that washout period. There are lots and lots of different trial designs, the crossover design, a factorial design. We didn't have time to include them all in today's presentation, all of them. We do cover some of this in one of the courses that we run, but it's a really good question. And up until about 24 hours ago, there were some slides in this deck that talked about crossover designs, and then we thought we would probably talk for too long when we took them back out.
Thank you very much. But I have a specific question concerning your work, particularly the one you're planning to go to the press on, the GBS3 trial. 320,000 participants. What sample size formula did you use to get that? I am looking at it, I can't just imagine how you arrive at that. I don't know, but I think I want to be educated on how you can get that large sample size.
I used to have just completely brown hair, as you could probably see from the photo, and now I have to dye my hair because the GBS3 trial has turned me completely grey. Partly because of that figure, but what that trial shows you is that actually patients want to take part in research. It's really, really incredible that 317,000 women have taken part in the trial, but they want to take part, and 85% of women in the UK want a test for Group B strep, and we don't offer it to them. And the reason they want to take part is that the approach we have currently is rubbish. It's imperfect, and the trial's going to give us the answer. But it's been a gargantuan effort, and it's taught me the importance of collaboration more than any other trial I've done, because we've needed people to pull levers to make it happen.
I guess the other thing to add about that, though, is that trial would not have been possible without the use of routine data. We would be recruiting forever. By the time we would have finished the trial, the question would have become obsolete potentially. So the use of routine data has been absolutely fundamental to doing that trial. We wouldn't just be grey, we'd be lying in a dark room, Yeah.
Please, the follow-up to that is whether you will be publishing with all the 71 facilities that contributed their data to your study. And that means the authors list will be another publication.
We've already got about 10 publications planned from that one trial. We've already published about 5, and there's another 10 planned. And it's led to so many additional studies. So we've worked with the UK Health Security Agency to look at colonisation rates by maternal ethnicity, and we're ready to publish that, which has never been done before because we'd taken all these swabs. We're working with the World Health Organization to time the update for their guidelines with the results of the trial. We've worked with a team in St. George's to help with developing a vaccine for Group B strep on the basis of our study. So, talking about research waste, you can also have research wealth. You can use all of these swabs for lots of different purposes, and all of this data can answer lots of different questions outside of the trial.
Thank you very much.
Thank you.
I'll take my seat now, but I will request that you take me back to clinical trial 101 by teaching me again what is meant by non-inferiority, non-superiority. What are you talking about?
Yeah, I'll happily have a conversation about non-inferiority.
Kamu, a professor of hematology and transfusion medicine at the College of Medicine, University of Lagos here.
Thank you.
Okay. Can I go with the next question?
Oh, okay. Thank you very much, Eleanor and Kate. Okay, I am Professor Mobolanle Balogun, Deputy Director, CCTRIS, for those that don't know me. My question is still boils down to the community engagement, patient and public involvement. You rightly said we do that, but not to the degree of ownership. Mm-hmm. And I'm thinking that our patients, our communities may not be as educated, research aware as what you have. And I know that you're probably dealing with, in the spirit of inclusivity. I heard you talk about, trying to include South Asians. And do you have tips for engaging, communities, patients that are not as enlightened or not as research aware, so that one can aspire to get to the level where you have this level of ownership that you described? Thank you.
That's a really great question. Thank you. The first thing to say is it's not easy. It's really hard. And any engagement or public involvement is better than none. So, what we've tried to describe is what we consider kind of exemplar, but, we know that it's really difficult to do. One of the biggest tips, I think, in terms of engaging with people who may be less familiar with research is to work with what we would probably term as grassroots organisations. So they might be groups of people, support groups, charities that are embedded in the communities who have established trusted relationships with the people they support. So if I were to walk into a room as a white researcher, middle class, reasonably well educated, into a group of people for whom I'd never met before, let's take the South Asian women that I was talking about earlier, and talked about my research, I'm pretty sure I wouldn't get very far. So I've worked very closely with people in the South Asian community who have already got established relationships with a wider group of people. So and they're already, when they're approached about the idea of the project, it's from somebody that they trust. There's a lot of fear, there's a lot of mistrust in research, understandably so. And so engaging in relationships with people that already have got that trust is really important. I would say we're still not quite getting it right in the UK. I think we're doing better than we used to do, but it's still a massive area of growth. So there are lots of women who may be less well educated, who are living in from much poorer backgrounds, who research is something that happens to over there, to some of the people. So we're working really hard to try and reach those communities better. I think in the past we've had an approach where we've expected them to come to us, and actually the researchers need to be going into the communities, establishing relationships over a long time. It's not a quick fix. I can't walk into a room, tell them about my project, get everybody enthusiastic and then walk away. You've got to establish relationships and maintain them over a long period of time. And I think that's starting to happen a little bit now, but it's slow. It's hard to do, but any progress is better than none.
Thank you very much, Prof. Eleanor and Prof. Kate. My question is on the umbrella trial. Does it mean that there is no control or comparator if we are testing multiple interventions in a particular disease or population? So is it safe to say that each intervention is a control to the other? That's my question.
Yeah, it's a good question, and I'll do my best to answer it. As I mentioned, we're certainly not experts in using the umbrella design at all. My understanding is that you would still have a control group. Often the control group might be current standard of care. and you're basically comparing with multiple doses or multiple treatments that are intended for the condition. Now, in a variation of that, using a platform or a MAMS design, the controls can change over time, because actually in the recovery example that Kate showed, dexamethasone, when that was shown to be effective, then became the new control, because clearly if you know something works, you shouldn't be withdrawing that from patients. So dexamethasone quickly
now. And I'm wondering what's the degree of harm and to what extent is the detriment of, maybe the antibiotic use compared to the clear evidence that, babies born with GBS, have, die and have so many complications. Is it really ethical? And I know they're waiting for RCT evidence, but then, and cost effectiveness. Sometimes I wonder if that cost effectiveness matter within the NHS is always, I don't want to say ethical, but it's always right. Because if there's some people that can afford it, for example, should that be an option? Is it? So, I mean, there some people question at the end of the day sometimes, but it's really something that bothers me for some of these issues. That's one. Can I respond to that?
CCTRIS in the UK, women who can afford a private test pay for one, and it's about £30 to have the antenatal culture test. So that's not acceptable. The group B strep support, the charity that campaign on this issue, have been lobbying the government for 20 years. And the barrier has been that the trial was thought to be undoable. So, the criteria set by the National Screening Committee that you have to have a randomised trial was just felt to not be possible, and it's because of routine data that it suddenly becomes affordable. Any paediatrician, any general practitioner is going to feel it's just wrong not to be testing women. And from my own personal experience, I was incidentally found to have group B strep, so I benefited from antibiotics. So I feel as passionately as you that we need to be addressing the question, getting the evidence. One other trial that Eleanor made me part was a modelling exercise that was done, which I'll show you. And it was quite extraordinary and the modelling exercise was done by the National Screening Committee scientists, but the number of women who would receive antibiotics was phenomenal. And that's what's led to a reluctance to adopt it. But I agree, when you have death and cerebral palsy on one hand, and some changes to a baby's gut microbiome, as a doctor, it seems outrageous.
Thank you. I believe one of the questions, my other main one is a comment about the one of the master protocol trials. So we finally completed PIPSICLE. Incidentally, PIPSICLE was our first trial, but we didn't complete it before the IVON trial because we're recruiting pregnant women with sickle cell disease, who are like 1% of the pregnancy population, and who you find maybe, 20 per site in over 2 years, for example. It was difficult. We eventually completed recruiting them in 4.5 years. And we've been accepted for publication in Lancet Global Health. So you see this. But the issue about PIPSICLE and why I feel that one of these master protocol trials, possibly the umbrella, because it's a rare condition. And before you can recruit so many different women for different interventions, it's going to be difficult. And there are several questions we still need to answer in them. So going forward, I feel that we'll probably need one of those in order to, especially since, for example, for PIPSICLE, at the end of the day, it was a negative trial. We didn't find effectiveness. And it's most likely because of the fact that, our numbers for the actual question were fewer than we expected them to be. However, we did find safety issues which we have to, look into going forward. How do we do that when, we're doing the routine type of trial?
So I think in trials with populations of rare diseases, utilising those alternative designs could be really fundamental, make it more efficient. And as you say, an umbrella trial might be appropriate. But there are also kind of more traditional designs, like the factorial design, where essentially you're answering multiple questions using the same population. So when you're studying a rare disease, and trials take such a long time, you really want to maximise efficiency. So, in a factorial design, you can potentially answer 2 or 3 research questions in one. And again, they're more methodologically challenging. You need to have experienced methodologists and statisticians who know that particular design really well. But there are ways, there are these designs that are more typically appropriate for rare diseases, which are just very difficult to recruit to otherwise.
Just at one point, with my sort of UK funder hat on, it's many people in the UK who work on rare diseases start their own registry. And just in, you would
I have 2 questions. You mentioned that we need more researchers. And I wanted to ask, how can students and early career professionals carry out and get involved in clinical trials? And also, how do you choose your research question? Because you've given us various designs and how to know which design to use for a particular intervention. But I wanted to ask, how do you know what to conduct a clinical trial on? Those are my 2 questions. Thank you.
Excellent questions. So, as we've talked about, they're complex and long and hard work, and so there's loads of work to do. So all of us working in clinical trials have more work than us and our teams can possibly do, so there's always opportunities. So the 10 studies I'm involved in have 10 medical students, 10 midwifery students, all working on different aspects. We have a medical student who's just done the focus groups of South Asian women with gestational diabetes to see what matters to them, what they need in the inclusivity package. So you just have to ask, you just have to, get involved, show willing, show determination, and then deliver it. And if you deliver it, they'll give you another project to do. So actually, you are in a wonderful situation where you've got CCTRIS here delivering really big, high-impact trials that is way more work than the poor CCTRIS team can do. So approach them, be confident.
Yeah, and I guess just to add to that is that, we've worked in trials for a long time. We learn new stuff every day. So every trial that we do, we learn something new. And that means there's not a kind of a typical pathway that people follow really in clinical trials, which makes it quite attractive to lots of different people. Kate and I have got very different backgrounds, but kind of complementary expertise. So work with people who you look up to, who you think are doing some great trials. And as Kate says, there will be opportunities for you to do different things. And the more trials you work on, the more experience you get. The first trial you work on, there's always going to be a first time, and then you'll just gain experience as you go. The question you asked about how do you come up with a research question is a really interesting one. So there are loads of ways you might do it. probably the best way is in some places there are existing lists of what are considered priority research areas. So, for example, I do a lot of work in preterm birth, and there is a whole list of about 15 very broad questions that are important to researchers, to health professionals, and importantly, families. So there's been a whole study that have identified these priorities, and rather than me thinking, oh, I'm really interested in a particular question, I go to this list and think, actually, we could try and answer this question because we know it's important to lots of different people. So I guess my advice would be talk to lots of people. So don't just think, oh, this is an interesting question to me. You've got to talk to your other peers, so other people working in the health system, try and talk to patients and find out, is this something that's of interest to them? When you come up with that, you could potentially find a research question that's absolutely key to changing something fundamental. But I think in the past, lots of research has been done on research questions that might be very interesting to the single person who's come up with it, but actually not terribly interesting to anybody else. And that again is research waste. So that's why collaboration is so important, right from the very outset, right from coming up with the question.
Thank you very much for this very interesting and lecture. I'm not quite my name is Ahmed Doloy, and I'm a basic medical science researcher. All the while I've been hearing about patient. So I'm wondering, would the study instead of non-intensive people, in fact, I'm actually interested in source sensitivity and source sensitive hypertension in people of African ancestry. So if I have a study that looks into redefining source sensitivity that involves high potassium diet intervention, I also try to look at the molecular.
I didn't quite get a bit about your diet. Can you just repeat a bit about the dietary?
Okay. I said I'm interested in source sensitivity. And African people of African ancestry, source sensitive hypertension. And when you talk about source sensitivity, it used to look at source sensitive, source resistant, but right now it's become more like a spectrum. So if I'm studying a group of low-intensity joint adults, and I intervene with the diet that is very rich in potassium to see if there will be a definition of source sensitivity in this group of subjects or people or participants. Can that be classified as a clinical trial?
Yes, it sounds like you're doing an experimental study, because you're going to do something to some participants and not do it to others. It depends whether you're planning to randomise, and therefore it would be a randomised clinical trial, or whether you're just going to implement the intervention in one group and then it's an observational study. Does that answer your question? Happy to discuss at the end. It depends whether you're planning to randomise.
All right. Time is fast spent. So I believe we still have questions, but we are not entertaining them here. All right. So I want to say thank you, but all right. Thank you for that. Thank you for that. So without much ado, we'll be inviting once again Dr. Okwade to kindly make the closing remarks while our professors make way back to their seat. Thank you very much. Dr. Okwade, please.
Thank you. I think we can do better than that. This is quite information packed, and I've learned quite a lot today. Thank you, Professor Kate Walker and Professor Eleanor Mitchell. That is quite outstanding. I will just stand on a existing protocol to give this a vote of thanks, but I will single out special recognition of the provost, ably represented by the Deputy Provost, Professor Daniel Debi, the Director of CCTRIS, Professor Falabi FAS, another distinguished personality that are here today. So, distinguished ladies and gentlemen, colleagues, on behalf of the College of Medicine, University of Lagos, and the Center for Clinical Trial Research and Implementation Science, CCTRIS, the trailblazing, the #1 clinical trial research center in the University of Lagos, University of First Choice and Nations. But I think CCTRIS deserve a round of applause. So let me use this opportunity to extend our deepest appreciation to our esteemed guest speakers. Again, Professor Kate Walker, thank you very much, Prof. Professor Eleanor Mitchell from the University of Nottingham, United Kingdom, for this, for their engaging and thought-provoking presentation today. I'm sure, just like as I've learned quite a lot, I'm sure every single one of us have taken away something here today. It is very important for us to acknowledge the fact that their expertise and generosity in sharing their experience with us today have really enriched our understanding of how to design and manage clinical trials more effectively. And of course, this will definitely position us as an institution to be able to carry out a very groundbreaking research going forward. Let me also use this opportunity to thank the Provost of the College of Medicine, ably represented by Professor Odebi, and of course the Dean, who I'm representing also, of the Faculty of Clinical Sciences, for their continual support for the center, and of course for providing leadership for the institution, and of course to all members of CCTRIS who organized this event. Let me single out Rachel, very wonderful work, Undubusi, Dr. Adara Moye, Wumi Adara Moye, and every Badmos, and every single member of the CCTRIS team that put together this very enriching event. And also to all participants, all the faculty, students, and guests, for your active engagement contribution throughout today's session. I think you are wonderful there, and I think you deserve a round of applause for yourself also. So it is our hope that the knowledge we have all gained here today will inspire stronger collaboration, better trial design, and of course, more impactful research that will be emerging from our institution, and of course in Nigeria, from Nigeria as a whole. Once again, thank you all very much for making this event a success. We look forward to welcoming you during our next seminar series. Thank you, and God bless.